Good afternoon, ladies and gentlemen. Thank you for the invitation. I'm just waiting for the sound to come on. I realize I'm last speaker, so I'm going to keep it very simple. So I'm talking about Rhizobia, the soil they live in, the soybean cultivars they interact with, and then also testing Rhizobia. So where do Rhizobia live? First of all, Rhizobia live as free-living microbes in the bulk soil in a non-plant associated manner. The moment you plant that soybean seed, Rhizobia in its direct vicinity will move to the rhizosphere. And then obviously at the appropriate time, in response to that flavonoid signal, the Rhizobia will move into the nodules and colonize the nodules. So in the bulk soil, the Rhizobia are saprophytes, meaning that they live on dead plant material. So obviously the native populations of Rhizobia, is the sound right? The native populations of Rhizobia depend on organic matter or plant material to live, and also the carryover inoculant from the previous season. The moment the Rhizobia is established in the rhizosphere, there's a unique dynamic going on. So the rhizosphere environment, in that environment, the plant takes care of the Rhizobia. So the Rhizobia lives off the root exudates. And obviously inside the nodule, as an endosymbiont, the plant provides sugars from photosynthesis to this nodule and to the Rhizobia inside the nodule. So our focus as inoculant manufacturers are the establishing Rhizobia in the rhizosphere. This quote is very interesting because we don't realize the complexity of the biological systems we work with. If you take a plant's rhizosphere, you can see it's a microbial hotspot and it's considered one of the most complex ecosystems on Earth. So this is really what we're working with. So there's no simple answers. Everything, I think the more we learn, the more we understand the complexity of this ecosystem. So just a quick overview of what the rhizosphere is. The rhizosphere, or the root interface, is literally that 5 millimeters of soil surrounding the root. So this is the area of soil that's under the influence of the root exudates. So these root exudates contain all the food for the microbes that live in this close association with the plant. And one of the first things microbes do when they establish in the rhizosphere is they produce a biofilm, a slimy substance that protects them and defines this rhizosphere. The rhizosphere has a unique pH, independent from the soil pH. You can see on this photo the soil pH being approximately 5.5, whereas the rhizosphere pH is highly alkaline. So this is really a chemically independent environment from the soil, and this is where your Rhizobia is established in that close interaction with the plant. The rhizosphere actually has, if you plant the same cultivar in the Western Cape, in the eastern part of the country, in Natal, it will have a very similar rhizosphere. But even in the same crop, I mean, there's subtle differences in the exudates. So there's actually crop-specific and cultivar-specific differences in the rhizosphere based on these exudates. So let's look at how Rhizobia interact with different cultivars. This was a question that we really had for a long time, and last year we decided, but listen, in one of these cultivar trials, we must start and evaluate the nodulation patterns. So this cultivar trial was planted close to Carletonville. We evaluated 5 soybean cultivars, 2 pioneers and 3 panners. The inoculant that we used 4 Rhizobia strains. 1 dose we put on the seed and another dose in furrow. So the strains we used were the WB74 and then 3 USDA strains. We didn't use any flavonoid inducers, so we relied fully on the plant to produce that flavonoid signal. And then we evaluated the nodulation patterns of these cultivars at R1 and R5. Oops. So there's a visual of what we've seen at R1 for the different cultivars. So you can see, so this is a summary of the nodules, the amount of nodules, and distribution from small, medium, large, and extra large on the different cultivars. So I don't want to go into too much detail, but you can see the first cultivar had an average of 41 nodules per plant, second one on average 1 per plant, the third one 8 nodules per plant, and then the last two between 1 and 2. But look at the size distribution. I mean, it's totally different between the cultivars. So that was really interesting to see that how different cultivars respond to exactly the same inoculant. At R5, the patterns look totally different. There you see now the first cultivar had only 7 nodules per plant, the second one no nodules per plant, and then the middle one on average 14 per plant, the last two only 1 per plant. What was more interesting for us was to see how many of these nodules were active. So obviously when a nodule initiates, it's a cream white color inside, and then during the active phase it changes from a light pink to a dark red color, and then the moment the plant inactivates that nodule, it becomes green, and then when it degrades it becomes black. So based on this color reference, we evaluated the trial based on the color of the nodules inside. So you can see all the cultivars that were still initiating new nodules at R1. The first one, 29 new nodules at R1. And there you can see in the red block all the active nitrogen-fixing nodules of the cultivars. What's interesting is look at how many of the cultivars already had inactivated nodules, the green ones, as well as nodules degrading. So from that 41 nodules per plant in the first cultivar, I mean, there was a significant amount, almost 50%, that was already degrading, that was not active and degrading. Look at R5. So there you can see the second cultivar, no active nodules, but look at the differences. I mean, the differences were just mind-blowing because we suspected differences, but I think not to this extent. And then obviously we all had to bet on our favorite who's going to win the yield race. So I must say my bet was on the third one because it was more the classical nodulation patterns. Yeah, that was what I expected. The first one was a little bit too good to be true, so I'm always cautious about too good to be true. So look at the yield comparison. So the one that actually won the yield was the one which had on average 1 nodule per plant. A very large nodule, but only 1 nodule per plant. I mean, in my scientific mind, that doesn't make sense. The classical nodulation pattern came into the second place, and then on the third place you can see the one with the large amount of nodules. So there's really something here that we haven't figured out. So this was an interesting article for me that talks about the process, the decision-making process of the soybean plant. So that is called the autoregulation of nodulation. So usually when I show producers this slide, they say, oh, I want the nodules on the right-hand side. I mean, it looks amazing. In this study, they mutated these plants to understand how the plant autoregulates the process of nodulation. So the plants on the right actually had like 270 nodules per plant. So it looks fantastic. But look at what happens in the field. So the first one on the left was where the plants weren't able to produce the flavonoid signal. The middle one was the wild-type genetics, where the plants senses nitrogen deficiency, they produce the flavonoid signal, the Rhizobia infect, and then that plant manages that nodule and the production of that nodule very carefully based on its energy status. The last one is that supernodulating strain, where you had those 270 nodules per plant. So those plants grow about this high and then they die. So it literally depleted that plant from all its energy. So very interesting dynamic. So this is my summary of how I see this process. I call the soybean plant the employer. And every employer, meaning every cultivar, has a different personality with different strengths and weaknesses. The moment that employer senses nitrogen deficiency, it will recruit employees, the Rhizobia and the Rhizosphere, to put up these nitrogen factories. So inside this nitrogen factory, the employer is very careful how he manages this factory based on stress, on its energy status, etc. So typical factors influencing this employer's decisions are nutrition, nutritional deficiencies obvious, like for example molybdenum and cobalt, stress, how that specific employer can cope with stress, and even biological stresses like nematodes, pathogens, and then abiotic stresses like heat, drought, waterlogging. Also, anything that happens with that plant that has an influence on that plant as the employer influences how he manages that nitrogen factory. And that's where the genetics come in. So different cultivars have different abilities regarding, for example, the nitrogen use efficiency. How that plant will convert nitrogen to amino acids to proteins. How that plant will convert phosphates to ATP energy. So this is really where the complexity comes in. So my conclusion is that we have more questions than answers. And working with this complex symbiotic interaction, we really need more insight in how different legume host cultivars interact with different Rhizobium strains to really unlock the full potential of this relationship. Rhizobium testing. So I don't know how many of you know that the technique that we're using today to test Rhizobia and to decide that it's a good quality or bad quality Rhizobia was developed by Robert Koch in 1883. So it's a really ancient technique, still used today. The basics of the technique is to dilute your sample 100 million times so that you can get a countable of amount of colonies on a plate. So first of all, if you dilute up to 100 million times, I mean, getting a really representative sample from a 1,000-liter fermenter it's a challenge. So I can say to you that this is not a precision technique. Researchers have described it at best as a gross estimation of cells per mil. The other challenge is obviously Rhizobia are very slow growers, so they take 7 to 10 days to get colonies on a plate. So it's not a quick test to give you that validation that you're looking for. And then obviously you need to validate your results. So you either have to validate it with counting chamber results, which are very laborsome. It's physically a live count of those Rhizobia under the microscope, or you have to use independent labs to verify your results. So this is just a statement about a scientist that's really involved in the validation of technologies, what he's saying about CFU or colony-forming units. CFUs is only an estimate of the number of cells present. It is a skewed estimate at best. Just to give you an indication of how skewed this estimate is. So this is inter-lab testing of Rhizobia CFU counts. So the same batch of Rhizobia sent to 3 different labs. We've all started the testing on the same day. So we personally delivered the Rhizobia in a cooler box to make sure there's nothing going wrong along the transport. So you can see the variation is, I mean, you actually don't know what to make with the results because, I mean, it can be a great product or it can be a product that's useless. So where do you position on this one? even in our lab, just intra-lab testing. And this is 3 technicians, all with science degrees, with extensive microbiology experience. So it's experienced technicians. Look at the variation just between 3 technicians doing the same counts on the same batch. So my question to you guys, and I want to put it out there, is how do we take Rhizobium testing to a new trustworthy level? I don't have the answers, but I know how to test that, to what criteria it must fulfill. So we need a more accurate test. It can't be too expensive because otherwise it won't be widely used. Preferably instant results, not waiting for results 7 to 10 days. The results must be consistent across labs. And this will really be an industry effort. I mean, we as a lab can't develop a test like this. And it must be accepted by all industry partners. So possible options to investigate is automated counters. We have tested a few in our lab, and the technology is becoming more and more reliable and precision, especially on the small bacteria like Rhizobia. Or otherwise, a rapid molecular test based maybe on the 16S RNA. So that was just options from my side that I want to put out there. I think there's enough researchers here that can maybe come up with much better options. I want to show you this. When I saw this piece of equipment, I was like, I wish we had this for Rhizobium testing. Imagine easy and fast Rhizobium testing that you can trust.
Cell culture almost always includes a step where cells are quantified. Whether the goal is propagation, cell health monitoring, or harvesting for experiments, it is almost always necessary to get an accurate count of cells from culture or primary tissue extraction. Chemocytometry has been used for decades, but is subject to errors in counting and calculation, not to mention that it's time-consuming and requires staining reagents that are often toxic. What if counting your cells was an efficient, integrated part of your cell passaging or collection protocol with Coulter counter precision? The Scepter 3.0 handheld cell counter gives you the cell counts you need when time counts in the culture room. Fast, efficient, precise. Each precision-manufactured sensor enables accurate culture impedance-based cell counts in seconds. Obtaining counts is as simple as pipetting. Simply insert the sensor into the sample and draw the sample into the instrument. Counts are displayed along with a histogram of the cell population by cell diameter or cell volume, as chosen by the user. The enhanced Scepter 3.0 counter design enables direct transfer of sample count data via the user's portable USB drive, or via wireless transmission from the Scepter 3.0 counter to the user's printer or computer workstation for analysis and archiving. The instrument stores hundreds of results for easy retrieval of archival data, and the charging station can be mounted anywhere, so the Scepter 3.0 counter is always ready to count when you are. Unlike other automated counters, the Scepter 3.0 counter requires no bench space. It needs no additional and potentially biohazardous reagents for operation, and no transfer of samples from the biosafety cabinet to sample loading on the bench. Meaningful cell culture requires accurate, efficient quantitation. Now, the smarter technologies you use to enhance daily efficiency have made their way into the culture room. Count on the Scepter 3.0 handheld cell counter for easy, accurate cell counts. Visit our website for more information and to order the Scepter 3.0 counter.
Yeah, I think that would be really nice. And that's all from me. Thank you.